Welcome everyone. Welcome to another video from Kundu Academy. Today I am going to talk about the pyrosequencing. Now, pyrosequencing is also a SBS technique. That means we are going to sequencing our template DNA by synthesizing its complementary strand, and it's a next generation sequencing. That means it is more advanced than Sanger sequencing. Now in this video I am going to talk about the principle or basics of the pyrosequencing. Then I will be talk about the type of the pyrosequencing. Then I will be talk about the reaction involved in this pyrosequencing techniques. And in last I will be talk about the efficiency of pyrosequencing. So let's talk about the basics of the pyrosequencing. As we know, in any synthesizing reaction or synthesizing of the DNA strand reaction, we need DNTPs, and we have the 3' OH in the existing complementary strand. Now, this 3' OH has a lone pair in its oxygen atom, and this lone pair going to attack the alpha phosphate of upcoming DNTPs, and as a result of that the PPI or pyrophosphate gets released and DNMP get incorporated in the complementary strand. Now from this pyrophosphate the pyrosequencing name came. Now this pyrophosphate undergo a cascade reaction. Now let's talk about what is the cascade reaction. Now let's say the three reaction we have from A to B in presence of the enzyme 1, then the second reaction is B turned into C in presence of the E2, and third one is the C turned into D in presence of the E3. As you can see, the product of the first enzymatic reaction act as a reactant for the second enzymatic reaction, and the product of the second enzymatic reaction act as a reactant for the third enzymatic reaction. So when the two or more enzymatic reaction connected to each other like that, then we call them as a cascade reaction. As we can see here, this is a cascade reaction. As a result of this cascade reaction, light produced, and by detecting this light with the help of the light sensor, we can detect the incorporation of the DNTPs. Okay, now talk about the types of the pyrosequencing. Now we have two types of the pyrosequencing. One is the solid and second is the liquid. In solid pyrosequencing, we need to remember a very important step, that is the immobilization of our template DNA. Why we need to immobilize our template DNA? I will be talking about it later. So, the solid pyrosequencing is a three enzymatic system. The enzyme involved in this is polymerase, DNA polymerase, sulfurylase, and last one is the luciferase. Now let's see the reaction of the solid pyrosequencing. Now, as the first step, we need to synthesizing our DNA. So, as you can see here, we immobilizes our template DNA, and we need to add the DNTPs sequentially, not four at a time. So, we need to add a single DNTP at a time in the presence of the DNA polymerase. As we all know, DNA polymerase we need for the synthesizing DNA strand, and from them the DCTP, as we have the G base in our template DNA, so DCTP get incorporated and the pyrophosphate get released, and these pyrophosphate interact with the APase in presence of the ATP sulfurylase and produces ATP and sulfate as a byproduct. Now this ATP interact with the luciferin in presence of the oxygen and luciferase enzyme and produces oxyluciferin. This is the oxyluciferin along with AMP and PPI. Now this oxyluciferin produces light. Now let's see how can we detect from that light this base get incorporated. As we go from downside to upside, what we get? We get the production of the light. Okay, light get produced. So what that means? That means the pyrophosphate get released. And what that means? That means the DNTP get incorporated in our complementary strand. Okay, now let's say why we need to immobilize our DNA. So think, in the reaction system we have our DNA template attached to it, that is immobilized. So when we add, let's say in that reaction we add DATP. Now DATP not going to incorporated in this reaction because we have G in our complementary strand or in our template. So we need to get rid of this DATP before we add any other DNTPs. So for get rid of this DATP, we need to wash our system. So if we don't immobilizes our DNA, that is our DNA template, if we don't immobilize it, so the DNA, our template DNA gets washed off in the washing step. So we need to immobilize our DNA. Now let's see the liquid pyrosequencing. Liquid pyrosequencing is a 4 enzymatic system. In addition of those 3, we have the apyrase as an additional enzyme. So what apyrase does? Apyrase is degrade unused DNTPs. Now in solid pyrosequencing we use washing step to get rid of unused DNTPs. Now in liquid pyrosequencing we use apyrase to do the same. So in liquid pyrosequencing we have some advantages, that is the no need for the washing, and in liquid pyrosequencing we run the whole reaction in a single tube. So now let's talk about the efficiency of pyrosequencing. Now as I told you before that pyrosequencing is a next generation sequencing, so the efficiency of the pyrosequencing is much more than the Sanger sequencing. In pyrosequencing, we can detect the incorporation of consecutive same bases or same DNTPs in our complementary DNAs up to 5 to 6 incorporation. So what that means? That means let's say this is our template DNA, and in this DNA we have 6 adenine at a stretch. So in complementary DNA there will be the incorporation of the 6 DNTPs. Okay, so the every incorporation of the DNTPs we can detect through pyrosequencing. So let's talk about the template preparation step. So for run the pyrosequencing reaction or any synthesizing reaction, we need to prepare our template. So in pyrosequencing, how can we do that? In pyrosequencing we run PCR. So as a PCR product we will get the amplified number of our DNA. Okay, now there is two problem in this product mixture. In this product mixture we will have some DNTPs, leftover DNTPs that are not used in the PCR reactions, and there is another problem, that is, let's say this is our template in the PCR reaction, and in this template the our primer of the PCR reaction will add in this manner, and from that position the complementary strand will synthesize. Okay, now we need to destroy this extra primer region. So how do we do that? So to do the both these job, we use three enzyme. Okay, now the first enzyme will be the alkaline phosphatase, or we can use apyrase. These two enzymes can degrade these DNTPs or destroy these DNTPs, and for this job we use exonuclease 1. So these exonuclease can destroy this region of our primer. For today, that's it. Thank you for listening me. Hope you enjoy the video. If you learn something from this video, please do like and please subscribe my channel for the upcoming videos, and feel free to criticize. And if you have any question about this video or any question about pyrosequencing, let me know in the comment box below. Thank you.